Hello dear students. In this lecture we will discuss mechanism of Agrobacterium mediated transformation. So Agrobacterium tumefaciens and Agrobacterium rhizogenes, these are soil bacterium which are found in soil, and when they infect plants they induce crown gall and hairy root disease respectively at the wound sites on dicotyledonous plants. And once they infect the plant and these tumors are formed, these tumors can grow and continue in the absence of this bacterium on tissue culture media or in nature also. So uh, these tumors are formed because the DNA of the bacteria or the agrobacterium gets integrated into host plant and results in the formation of unlimited supply of auxins and cytokinins, which results in tumor. So the tumor tissues also synthesizes novel amino acids and sugar derivatives, which are known as opines, and the bacteria utilizes these opines for their growth. So the strain of Agrobacterium, it synthesizes different kinds of opines which are called octopine and nopeline. So here you can see in the picture, the first picture is of Agrobacterium tumefaciens, which is showing a TI plasmid, pink color TI plasmid. So this TI plasmid has a region called tDNA which gets integrated into the host plant when the bacterium infects the host plant and causes tumor. So this tumor is caused because the TI plasmid has a special region tDNA which has genes for auxins and cytokinin synthesis, and it starts synthesizing unlimited supply of these hormones. So these results in the formation of tumor, and it can occur even in the absence of bacteria. So the second picture is of tumor which is called crown gall, which is formed by Agrobacterium tumefaciens, and the picture at the bottom, it is showing the hairy roots which are caused by Agrobacterium rhizogenes. So, what is the mechanism of um transferring its tDNA into host genome of the plant by Agrobacterium? So, you see in this text that there is a host explant and there is Agrobacterium. So, this Agrobacterium has a TI plasmid, and the TI plasmid has a region called tDNA. This tDNA has some genes which are responsible for the formation of auxins and cytokinins. These are called oncogenes. So these oncogenes are replaced, and they are replaced with the foreign gene of interest. So this replacing of oncogenes is called disarming. So here in this text you can see that the host explant plus disarmed agrobacterium, when they come in close vicinity, when the host explant is wounded, there are specific chemicals called acetylseringon. They come out from the wounded cells of the host and they attract agrobacterium. So this process is called chemotaxis. It activates the Vir genes which are present on TI plasmid, and the Vir genes, they act and cut the tDNA part. By unknown mechanisms, the tDNA, it gets integrated into the host genome and leads to the development of transgenic. So this is the mechanism of gene transfer, that is tDNA or transfer DNA, how it goes and gets integrated into host plant. That is why the Agrobacterium tumefaciens is also called natural genetic engineer, because It is naturally the tDNA is transferring from Agrobacterium tumefaciens to host plant. So the tDNA, the Ti plasmid, which is present in Agrobacterium tumefaciens, it has four major regions, which you can see here. Vir region, which is called virulence region, tDNA region, which has genes for auxins, cytokinins, and opine synthesis, and the tDNA region is has border and right border, which are made of 25 base pair repeats. Along with this, there is origin of replication region, and there is opine catabolism region. So these are the four regions of Ti plasmid, tDNA, Vir region, Con region, and Ori region, which have different roles to play. And you can see here that Vir region has five genes, more than five genes: Vir A, Vir B, Vir C, Vir D, Vir E, Vir F, and Vir H, which have different roles to play during transformation. So Vir A is results in the formation of a chemoreceptor. Vir B and C, they have unknown role, and they probably have a role in extranuclear transport of tDNA. D encodes restriction endonuclease to cut tDNA. E helps the tDNA to integrate in plant genome, and Vir G, activates the rest of the Vir genes. So, in the flow chart, you can see that the Vir A gene of the virulence region, it encodes a chemoreceptor which combines with the acetylserinone released from the wounded host cells. So this complex of Vir A chemoreceptor and acetylserinone activates Vir G gene. Vir G gene activates rest of the virulence genes, which are B, C, D, E. Then Vir D encodes endonuclease, which forms NICs at 25 base pair borders. It means that Vir D forms endonuclease, which cuts the 25 base pair borders of the tDNA, and because of this, the single stranded tDNA comes out, and by the action of Vir E, the tDNA goes and gets integrated into plant genome. So after cutting the tDNA, or after nicking, the tDNA forms a complex with the protein Vir E and gets transported to plant nucleus. So the other genes like Vir B and Vir C helps in extracellular transport of tDNA into host genome. So the natural this is how the transfer of tDNA occurs from Agrobacterium tumefaciens to host genome naturally when the Agrobacterium infects the wounded host cells. That is why Agrobacterium tumefaciens is called natural genetic engineer. And this property is exploited by scientists. This property of Agrobacterium tumefaciens is exploited by scientists to transfer the gene of their own interest, which are called foreign genes. So here you can see that tDNA is present in the Ti plasmid, and this tDNA we have just discussed. They have the genes for auxin, cytokinin synthesis, as well as opine genes. So when the Ti plasmid is genetically engineered by tools and techniques of biotechnology, these are replaced by the gene of interest and the gene of selectable marker and reporter genes. So then you can see that the picture at the bottom shows the co-integrated plasmid, which has a virulence region, origin of replication, and tDNA, where the oncogenes that are replaced with the gene of interest, as well as reporter and selection marker genes. So now this is a engineered plasmid, and this plasmid having the foreign gene of interest, like foreign gene of interest can be any gene like disease resistance gene, virus resistance gene, abiotic stress tolerance gene, any gene, bt gene. It will be transferred to plant cell artificially via by co-cultivation with this genetically engineered Agrobacterium ti plasmid. So this is how the mechanism of gene transfer takes place in tissue culture lab. So we have a TI plasmid and we have a gene of interest. These are ligated together to form a recombinant TI plasmid, which is your genetically engineered TI plasmid. This is inserted in Agrobacterium, and then this Agrobacterium is co-cultivated with the host plant, and then by the natural mechanism the tDNA gets transferred. The tDNA with our gene of interest get transferred to the plant, and the plant is a transgenic. So this is another picture which is showing that first step, where you see that the Agrobacterium tumefaciens ti plasmid having tDNA and uh, the DNA containing gene of desired trait, they are combined to form a recombinant TI plasmid. In the second step, it is introduced in the plant by co-cultivation with Agrobacterium, and the plant, the explant of the plant gives rise to a new plant by the totipotent nature of the explants in tissue culture media. So this is again the same picture, and this is another picture where you see that there is a tobacco leaf. From this, the leaf discs are cut, and they are co-cultivated with Agrobacterium for 24 hours on a selection medium, which allows plant cells that have acquired DNA from bacteria to proliferate. Then they form shoot, then they form root, and it results in the formation of the whole plant. So this was the mechanism of Agrobacterium mediated transformation, and there are many advantages of this transformation technique. This is a natural method of gene transfer. Agrobacterium can conveniently infect any explant. Large fragments of DNA can be efficiently transferred. The stability of transferred DNA is good. Transformed plants can be regenerated effectively, but there are some limitations also, like there is a limitation of host plant of Agrobacterium. It by and large it does not infect monocots, and the cells that regenerate, they are more efficiently often difficult to transform. So agrobacterium mediated transformation is the most common method used for making transgenics or genetically modified plants, and I hope the topic is clear to you. Students, please refer to both lectures 1 and 2 of agrobacterium mediated transformation to make your concepts clear, and please refer books to to for a better understanding. So thank you, students, and let us meet in next lecture.